Hi, good afternoon, everybody. My name is Brian Kane. I'm the publisher here at Xconomy. As I hope you all know, Xconomy focuses on engaging life science leaders and innovators at the intersection of science and technology. Our mission via our daily newsletters, accelerating life science events, and award programs is to connect the funding, partnerships, technologies, and science necessary to support and enhance the successful development of novel therapeutics. Today's presentation, Simple Signaling Reporter Assays, Easily Quantify Activation and Inhibition of Cellular Pathways, is brought to you by Eurofins DiscoverX, who are committed to accelerating multimodality drug discovery and development programs from discovery to QC lot release. Today, we'll introduce you to the latest cell-based reporter gene assays designed to study signaling pathways. We'll discuss assay applications to quantify activation and inhibition of several pathways, and we'll provide you with a comparison of functional data from two cell-based assay types, distal reporter-based assays and proximal receptor signaling assays. Just a couple housekeeping items before we begin. If you have any technical difficulties during today's session, please press the Help button on your player console to receive assistance in solving common issues. Now, this is an interactive event, so we welcome all of your questions. In order to submit a question, simply type it into the question window on the left-hand side of your screen and then hit the Submit button. We'll be answering as many questions as possible during the Q&A session following the main presentation, but please feel free to send in your questions at any time and we'll add them into the queue. If at any time you're having audio difficulties or advancing of the slides, simply hit your F5 button to refresh your webcast console. And please be aware that today's session is being recorded and will be available on Xconomy's website, xconomy.com, beginning tomorrow for your review. You will receive notification by email when the on-demand presentation is available. So without further delay, I'm happy to introduce today's presenter, Dr. Jennifer Lynn Jones. She's the Senior Group Leader of Assay Development at Eurofins DiscoverX. Jennifer, the floor is all yours.
Thank you. In this presentation, the Eurofins DiscoverX portfolio of reporter assays will be described. These cell-based reporter gene assays have been developed to expand our menu of cell-based assays that allow customers to better understand how their candidate therapeutic molecules affect specific signaling pathways. Signaling pathways are a complex series of events that affect a cellular function, usually by changes at the transcriptional level. Some examples of cellular behavior mediated by signaling pathways are cell survival or cell apoptosis, increased or decreased cellular proliferation, and cytokine production by the cells. Shown on the right is one example, the signaling pathway for the NF-kappa B transcription factor, labeled as P50 in this diagram. Signal pathway initiation typically occurs at the plasma membrane with a receptor activation event and proceeds through a series of cellular events, ultimately resulting in changes in transcription in the nucleus that in turn modify a cellular function. Drug developers have generated therapeutics that can target different proteins or signaling events at any point along a pathway. These drugs are designed to alter specific cellular functions. Reporter pathway assays measure effects of therapeutics on an event near the endpoint of a signaling pathway and therefore can monitor effects of test compounds that affect steps almost anywhere along a signaling pathway. Reporter genes have long been used to identify important transcription factors and regulatory transcriptional elements, as well as to measure signaling pathway activity. A reporter gene is comprised of a transcriptional response element that normally drives expression of a target gene in the pathway, but has been engineered here to control expression of a reporter protein. When pathway signaling is activated, a change in reporter gene transcription and translation occurs. These effects can be measured by monitoring reporter protein levels or reporter protein activity. Some commonly used proteins used for reporter genes are luciferase or fluorescent proteins, since they are readily and easily measured. Thus, the abundance of a reporter protein represents a direct measure of signaling pathway activity. Effects of therapeutics modulating pathway signaling can therefore be evaluated by measuring changes in reporter protein expression. In the following slides, I will describe how we have developed reporter gene assays using the well-established Eurofins DiscoverX enzyme fragment complementation technology. So shown here is our enzyme fragment complementation technology, or EFC as we call it. And it's based on a split beta-galactosidase enzyme system. So the beta-galactosidase enzyme is divided into two fragments. One is the smaller enzyme donor, or ED fragment, and the second is the bulk of the beta-gal enzyme, or the enzyme acceptor, or EA fragment. Individually, each of the fragments has no enzyme activity, but when brought into proximity or mixed together, the two enzyme fragments can complement to form a functional beta-gal enzyme whose activity is readily measured by the addition of luminescent substrate. For our reporter gene assays, stable cell lines have been generated that contain a transcriptional response element controlling expression of a reporter protein tagged with a smaller beta-gal enzyme fragment. In the diagram on the right, this smaller enzyme fragment is labeled enhanced pro-label, or EPL for short, but we use this term interchangeably to describe the ED, or enzyme donor enzyme fragment shown in the diagram on the left. EPL tagged reporter protein expression is a function of signaling pathway activity and is measured by adding the complementary EA enzyme fragment and enzyme substrate to detect enzyme activity. The advantages of using the EFC system is it's just a well-established system that we've been using quite a long time for developing cell-based assays. It's a very simple homogeneous mix and read assay format and is easily scalable for high throughput screening. So shown on this slide are the first examples of our pathway reporter assays, and they utilize a reporter gene for the NF-kappa B pathway, which I showed earlier in the introductory slide. Assays with this reporter gene have been developed in two different cell backgrounds using two different activators of the pathway. So we've developed this in the U2S background shown on the left and the HEC293 background shown on the right. And the U2S cell line utilizes the CD40L as ligand, whereas the HEC293 uses TNF-alpha as a ligand. These ligands are used to activate receptors endogenously expressed by each cell type. TNF-alpha can activate the reporter gene in both assays, and the data in the HEC but only the U2S cell line responds well to CD40L, as shown in the graph on the left. The difference in the response to CD4L in the two assays is likely a consequence of receptor expression profiles for each cell background, where the HEC293 exhibits little to no CD40 receptor expression. Thus, reporter genes can be tailored for specific activators of a particular signaling pathway by the choice of cell background used for the assay. Alternatively, cells can be engineered to express the target receptor of interest to activate the pathway, and this is what we did to develop an assay for rank using the NF-kappa B reporter assay. So this slide shows our rank reporter assay, and again, it was created by rank gene transfer into the U2S NF-kappa B pathway reporter cells. And from this combination, we were able to develop a cell line that responds very sensitively to rank L. The EC50 for rank L is in the nanogram per mil range. The graph on the right shows results from testing with the therapeutic rank L inhibitor Prolia, demonstrating the potent inhibition of rank activation in response to rank L. Prolia is currently used to treat osteoporosis. So in this slide are results from another of our reporter gene assays that utilize endogenous receptor, the HepG2 STAT3 reporter assay. In this assay, IL-6 is used to activate the STAT3 pathway in HepG2 cells. Recently, the contribution of IL-6R pathway signaling to outcomes for COVID-19 patients has been documented. Elevated IL-6 levels have been observed in COVID-19 patients. Tocilizumab, an immunosuppressive drug currently used to treat rheumatoid arthritis, has recently been approved for phase 3 clinical trials of COVID-19 patients that have developed pneumonia. Data on the right shows results using tocilizumab inhibition of IL-6 activation of the STAT3 reporter. These results with tocilizumab demonstrate the potential utility of this reporter gene assay for the development of new therapeutics. Another reporter gene assay presented here measures T cell activation occurring through the T cell receptor. This assay provides a much easier and quicker alternative to more traditional methods of measuring T cell activation by proliferation assays or cytokine secretion. Here, a potent CD3 antibody was used to activate the JERCAT NFAT reporter genes by pre-coding increasing concentrations of the antibody on assay plates, followed by NFAT reporter cell addition. So that data is shown in the graph on the right. The data on the right demonstrates the assay in antagonist mode when a small molecule inhibitor of protein kinase C was used to inhibit the signaling pathway occurring downstream of a receptor activation. This reporter gene is another example of an assay that can be used as a building block to further modify the assay so that other cellular signaling inputs affecting the pathway, in this case T cell activation, can be assessed. So here I'm giving an example of how we use that JERCAT NFAT reporter assay, which measures T cell activation, to develop additional assays. So the immuno-oncology checkpoint receptors have been targets of intense drug development due to their therapeutic value in stimulating the immune system to combat cancer. Immune checkpoint receptor activation results in the inhibition of T cell activation and suppresses the immune response. One such receptor is PD-1. Here we demonstrate how the JERCAT NFAT reporter assay cells were further modified to generate an assay for the PD-1 checkpoint receptor. So I showed in the previous slide that with T cell activation, we see an increase in NFAT reporter activity. But to develop these assays for these checkpoint receptors, we've used a co-culture system instead of using the CD3 receptor antibody to activate the pathway. When the reporter assay cells and increasing concentrations of activator cells, so in this case U2OS cells expressing a TCR activator on their plasma membrane, when these two cells are co-cultured together, we see a large increase due to TCR and T cell activation. When PD-1 is expressed in the reporter gene cells, is co-expressed with the reporter gene in the JERCAT cells, and that curve is shown in blue, even in the absence of the addition of the PD-1 ligand to activate, we see a decrease in the amount of TCR activators and TCR activation and T cell activation, resulting in a reduced amount of NFAT activation. When the PD-L1 ligand is added to the ligand cell line or the activator cell line, the U2S TCR activator cells, and this curve is shown in red, there's a further reduction in the amount of T cell activation observed, and therefore a decrease in NFAT activity. So what we've shown is that by adding the PD-1 receptor to the reporter gene cells and the PD-1 activating ligand to the activator cells, or the ligand activator cells, we have developed an assay to screen for therapeutic molecules that can release PD-1 inhibition on T cell activation. So this schematic illustrates how the PD-1 NFAT reporter assay works in greater detail. The assay consists of two-cell co-culture system where one cell line is used to present ligand and activate the signaling pathway. These are the U2S cells co-expressing the T cell activator and the PD-L1 ligand. So when cultured together, we get both an activation by the T cell activator and PD-L1 activating PD-1 on the JERCAT NFAT reporter cells. So when the two cell lines are cultured together, some T cell activation is measured as a consequence of TCR activation of the T cell receptor, but as shown on the previous slide, it is reduced compared to the co-culture of cells without PD-1 and PD-L1 co-expressed. So the PD-L1 activation of PD-1 gives some inhibition of the T cell activation. Okay. So in this slide, we use this assay to screen for antagonists of PD-1 signaling. So in this case, an anti-PD-1 antibody is added to the co-culture system. And in this case, the PD-1 antibody shown on the right is blocking the interaction of PD-1, PD-L1 interaction and blocking the inhibition of or attenuation of the T cell activation. As a result of this loss of inhibition, greater T cell activation is observed. And so what we see is a gain in signal in this assay with this anti-PD-1 antagonist. In this schematic, I've shown the basics of the PD-1 signaling pathway. And what I'd like to point out is that we've also developed a PathHunter assay which measures a different event in the PD-1 signaling pathway. So shown on the right in the boxed in purple and towards the bottom of the slide is the PathHunter JERCAT PD-1 NFAT Pathway Reporter Assay that I've been describing. So it measures NFAT activation, which is a direct measure of IL-2 production. So it's a very downstream event measuring transcription, whereas our PathHunter JERCAT PD-1 signaling assay, which is boxed in blue on the left, measures a very early proximal event occurring close to the time of PD-1 activation by the ligand. This PathHunter JERCAT PD-1 signaling assay is actually so upstream that it measures PD-1 activation independent of TCR activation prior to when PD-1 activation has its effects on T cell activation. So we're able to assay for two different points of the PD-1 signaling pathway, both a proximal event and a distal event. So just to describe the PD-1 signaling assay that was described in the previous slide, I've presented this schematic here to show how our PD-1 signaling assay works and how it works independently of T cell activation. Like the PD-1 NFAT reporter assay, this assay is also a co-culture system where ligand cells are used to activate PD-1 in the signaling cell line. JERCAT cells express a full-length PD-1 receptor, but this receptor has been engineered with the small beta-gal enzyme ED donor fragment fused to its C-terminus. And this fusion protein is co-expressed with SHP1 or SHP2 phosphatase fusion protein, and these phosphatase fusion proteins contain the complementary beta-gal or EA enzyme fragment. So ligand engagement through co-culture with the ligand presenting cells So the PD-L1 engagement of the PD-1 receptor results in tyrosine phosphorylation of the PD-1 checkpoint receptor PK fusion protein in the cytosolic ITIM motif of PD-1. And this in turn leads to the recruitment of the SHP1 through the SH2 phosphotyrosine binding domain of SHP1 or SHP2. As a result of this recruitment, the two enzyme fragments are brought into proximity, and complementation of the enzyme fragment occurs and results in an active beta-gal enzyme. This active enzyme hydrolyzes substrate to create a chemiluminescent signal as a measure of receptor activity. So again, this assay is designed to screen for antagonists of PD-1 signaling. And so in the case of adding an antagonist PD-1 antibody that blocks the PD-L1, PD-1 interaction, no complementation occurs as PD-1 is not activated and does not recruit the SH2, the SHP1 EA fusion protein, and there is a loss of signal. So I've presented two assay formats that both measure PD-1 signaling the PD-1 signaling pathway, just different events, either a proximal event very close to the receptor activation or a distal event, which is very close to the endpoint of the PD-1 signaling pathway, which results in changes in transcription. So both PathHunter PD1 assay types provide a comprehensive understanding of the PD-1 pathway and different modes of action of the anti-PD-1 antibody. So the results shown on the left are the results from the PathHunter PD-1 signaling assay, in which blocking with a PD-1 antibody results in a loss of signal, as there is a loss of the receptor activation and loss of its recruitment of the SHP phosphatase to the phosphorylated activated receptor. In contrast, the PathHunter DurCap PD1 NFAT Reporter Assay is also measuring T-cell activation, whereas the PathHunter DurCap PD1 signaling assay works independently of T-cell activation. So in the case of the DurCap PD1 NFAT Reporter Assay, which is measuring a very distal event, it's measuring the combined inputs of both T-cell activation and PD1 signaling, which results in inhibition of the T-cell activation. So when the PD1 signaling is blocked, it releases the inhibition by the PD1 signaling pathway, and we get a greater increase in T-cell activation and NFAT reporter activity, and thus we get a gain of signal. Both assays exhibit quite potent responses with the anti-PD-1 antibody in the 100 nanomolar range, despite the fact that they're measuring two different events in the PD-1 signaling pathway. The PD-1 signaling activity is a more direct measurement of PD-1 receptor activation independent of the T-cell receptor, whereas the PD-1 NFAT reporter assay is measuring the combined effects, as I mentioned earlier. So in this slide, I've compared the two signaling pathway assay types. So what we've been describing are reporter gene assays in the column on the left, which measure a very distal pathway readout. And this contrasts non-reporter gene assays, which measure usually events in the pathway that are upstream of transcriptional changes. So the reporter genes, since they are a very distal event in a signaling pathway, they can represent the cumulative effect of signaling from multiple inputs modulating a response. So for example, it can monitor effects on both T-cell activation or TCR activation and PD-1 inhibition, since the transcriptional changes resulting from the pathway signaling pathway are measured. Therefore, results with reporter assays represent the cumulative effects from a signaling pathway and cellular response and can monitor different inputs affecting the pathway, as well as effects on many different steps in a pathway. In contrast, non-reporter gene signaling assays typically measure a specific event in a pathway upstream of a transcriptional effects and can more directly affect the mode of action of a drug. Both assay types, as illustrated by our two complementary PD-1 signaling assays and NFAT reporter gene assays, yield results with robust signals and signal to background ratios and demonstrate good potency with therapeutic molecules. Both assays have short assay times and are simple and easy to run. The PD-1 signaling assay is already available in bioassay format for QA applications. Thus, the two formats provide customers a choice in how they would like to test their compounds using two different means of measuring their therapeutic molecules. So finally listed here are the current list of assays that we offer using this reporter gene format. And also provides a link on the bottom below the table that provides more information about our reporter assays. I'd also like to point out that we offer a custom assay development service to develop new reporter gene assays or to modify existing assays to fit a customer's specific target or need. This service takes advantage of our cell-based development and cell engineering expertise. Cell-based assays are valuable tools throughout the drug discovery process. However, internal assay development can be challenging, expensive, time-consuming, and require many months to develop. The email for CAD inquiries is listed on the bottom right side of the slide. So in this last slide, I would like to summarize my talk by reviewing the advantages of our PathHunter Pathway Reporter Assays. These assays take advantage of our well-established enzyme fragment complementation system that are quick and easy to run. It's very simple. Plate the cells, add test compounds, add detection reagent all in one well, and read. Rapid responses result compared to the more traditional phenotypic cell assays for assaying a cell response. I've showed validation data with our reporter genes that demonstrate the specificity and sensitivity of the assays, as well as how the assays have been used to test both small molecule inhibitors and biologics. The addition of reporter gene assays to our already expansive portfolio of pathway assays with alternative formats give an alternative readout at the distal part of a signaling pathway. I have presented data from both our PD-1 NFAT reporter assay and PD-1 signaling assay as an example of how assays that monitor either a more receptor proximal or distal event like a reporter gene can provide complementary results for drug discovery. And finally, these reporter gene assays not only provide an off-the-shelf tool for evaluating drugs, but also as a building block for development of additional pathway assays. I'd like to thank you for your time and attention today.
Okay, great, Jennifer. Thank you very, very much. We do have a few questions in the queue for you. To the audience, if you do have any questions on your mind, now is the time. And while our presenters are answering, or while Jennifer is answering your question, it's a great time to complete the feedback form that appears on the bottom of your screen. So Jennifer, the first question that we have from the audience, what are the pros and cons to using the PD-1 signaling assay versus the reporter assay?
Thanks, Brian. So I did try to go over this during the presentation, and I think each of the assays has their advantages depending on what the customer is interested in, and what their priorities are. So for the signaling pathway, I mean, some of the advantages are that the assay has a very short incubation time, especially compared to more phenotypic assays. So the incubation time for the co-culture is actually only 2 hours. And you can see, even with such a short incubation time, that you get a very large, robust response with a large signal-to-background ratio. The pathway assays are slightly longer. They're an overnight incubation. But again, it depends on what the customer wants. The signaling pathway is, much closer to the receptor. It interrogates what's going on at the level of the plasma membrane. So it may be more appropriate for customers that aren't really interested in understanding how their molecules directly affect the pathway. Whereas the reporter pathway is closer to the distal end, as we emphasized during the presentation, and represents is probably more representative of how a drug treatment will affect the cellular behavior. And it can also take into account all the inputs that are affecting a pathway rather than just something that's occurring at the cell at the receptor level. Okay.
Got it. Great. Yes. No. Perfect. Thank you. Thank you very much. Another question we have from the audience: How do the pathway reporter assays compare to other commercial cell-based reporter assays?
Yeah, thank you, Brian. It is true that there are a lot of companies offering reporter gene type assays, and as I mentioned before, this technology has existed for quite a while now. And so I think, again, while we don't have direct comparisons between our assays and other assays that are available. I mean, I've sort of surveyed what the data that's online, and our assay results seem to give similar sensitivity to most others on the market. And again, like most reporter assays, they give very robust responses. The signal to background ratios are very large, and they're sufficient enough to serve a customer's purpose for investigating any of their targets. So, yeah. Okay.
Yeah. No. That's great. Thank you for that. And we know there's a lot of work and development being done, in this area. So just in general, what other reporter assays are currently in development?
So we have right now we have a handful of reporter genes in development. So we have one for the interferon gamma, and then the ROR gamma T, and also an ELK1 reporter gene assay, which we're trying to develop for the FGF receptor pathway, and then also the SMADs for the TGF beta pathway. But again, I want to emphasize that, we have a very strong custom assay development team and track record. So any reporter gene assay that would be of interest to the customer, we're definitely open to doing that assay development.
Great. Okay. Terrific. Well, to our audience, if we're going to have to cut it off right now, just based upon the time. But if you do have any questions, Dr. Jennifer Lynn-Jones and the team there at Eurofins Discovery X are available for you to answer the question. You can contact and get in touch with them directly. I would like to thank you, Jennifer, and the team there for an outstanding presentation, and to our audience for your questions. Again, if we didn't get to your question, Jennifer and the team will be happy to get back to you directly. But before you go, if you could please take a moment and answer the feedback form that appears in the bottom of your screen, that would be very helpful to all of us here. So thank you very much again, and on behalf of Xconomy and Eurofins Discover X, we hope you have a productive remainder of the day. Take care and stay safe, everybody. Thank you.